the prediction engine. You are not experiencing this moment. You are experiencing your brain's best guess about this moment, assembled slightly before it arrived and edited afterward to feel seamless. The thing you call now is a forecast, and the thing you call anxiety may not be a response to anything happening in your life at all. It may be a habit your brain learned so well it forgot how to stop running. Lisa Feldman Barrett at Northeastern University spent decades analyzing neuroimaging data and found something that overturns the basic story most of us carry about how feelings work. The brain does not wait for sensory input before generating emotional responses. It runs continuous predictions about what the body will need next, then updates those predictions when reality diverges. Her 2017 book, How Emotions Are Made, synthesized this into a formal model. It drew on the earlier predictive processing framework developed by Carl Friston at University College London, a theory so comprehensive it has been called the most important framework in modern neuroscience. It has been cited over 12,000 times since its publication in Nature Reviews Neuroscience in 2010. The numbers alone are staggering. The brain accounts for approximately 20% of the body's total energy budget, despite comprising only 2% of body mass, and according to Marcus Rachel's 2010 research, most of that energy goes not toward processing incoming sensory information, but toward maintaining predictive models. Your brain is spending most of its resources on its own forecasts, not on the world in front of it. David Eagleman's work, published in Incognito in 2011, puts an even finer point on it. The brain's reconstruction of now runs approximately 80 to 500 milliseconds behind real events. You are always living slightly in the past, watching a film your brain assembled just before you arrived. What this means in practice becomes clear in Alaya Crumb's research at Stanford. In one study, participants were given the same milkshake, 380 calories, but told it was either an indulgent 620-calorie treat or a sensible 140-calorie option. The body produced measurably different ghrelin responses, the hunger hormone, following the label rather than the contents. In a separate study, Crumb worked with Peter Salovey and Sean Acker and published in the Journal of Personality and Social Psychology in 2013 that a 2-minute mindset shift video shown before a stressful task produced a measurable shift in participants' physiological stress profile. The body's response followed the prediction, not the objective situation. Barrett's own research on what she calls emotional granularity adds another layer. People with larger emotional vocabularies, more precise predictions available to the brain, show measurably less amygdala reactivity to identical stress stimuli. The precision of the model changes its output. Mainstream science would say this makes sense within a well-established framework. The brain processes sensory input, computes an appropriate response, and generates an emotion as a readout of that computation. Anxiety is a learned response to perceived threat patterns. The brain is a sophisticated input-output system, and emotional dysregulation reflects errors in the output stage. But the timing is wrong. Emotional and physiological responses register in the body before the conscious mind has finished identifying the trigger. If the response consistently precedes the completed perception of the stimulus, it was not a response, it was a prediction. And if the brain predicted wrong, if it learned a threat pattern from an older, more dangerous version of your life and never received a formal update, it will keep generating that forecast indefinitely, regardless of what is actually happening now. What if chronic anxiety, depression, and emotional instability are not disorders of feeling, but disorders of prediction? What if the brain is accurately modeling a situation that no longer exists? And if emotions are forecasts rather than reactions, does that mean the path through them is not management or suppression, but the slower, stranger work of giving the brain new data until it finally revises the model. Some researchers believe that is exactly what is happening. We may never have a complete answer, but the question itself changes everything downstream. If the brain is predicting, it needs data. And there is one source of data that almost every person on Earth has been taught explicitly or implicitly to ignore. The sense that doesn't have a name in your language. A teacher probably had you count your senses on your fingers at some point in your life. Sight, hearing, touch, taste, smell. 5, tidy, complete, sufficient. But there is a sixth, not metaphorical, not paranormal, a documented neural system with its own dedicated pathway that monitors the internal state of your entire body in real time and feeds that data directly into every decision you make. It has a name. It is called interoception. And the reason most people have never heard it is not because it is obscure. It is because what it explains is enormous. Antonio Damasio at the University of Southern California spent decades studying patients with damage to the ventromedial prefrontal cortex, the region that receives and integrates interoceptive signals. His landmark patient, known as Elliot, had surgery to remove a frontal lobe tumor that damaged this region. His IQ tested above 130. His memory was intact. His logical reasoning was measurably unimpaired. But Elliot could not make decisions. Simple choices, which appointment slot to take, which pen to use from a cup on a desk, required hours of unresolved deliberation. Damasio published his conclusion in Descartes' Error in 1994. Decision-making is not a purely rational process. It requires the body's emotional signal as a navigational input. Without the gut, the brain spins. The architecture of this system is more elaborate than most people realize. Interoception travels on its own dedicated neural pathway, the lamina 1 spinothalamo-cortical tract, projecting from every organ in the body to the insular cortex, where it generates what we experience as gut feelings, hunches, and somatic emotion. Sarah Garfinkel at University College London published research in Nature Neuroscience in 2016 showing that people with higher interoceptive accuracy experience measurably reduced anxiety in response to identical threat stimuli. The stronger the body signal a person can consciously access, the calmer their behavioral response to the same objective pressure. Hugo Critchley at Brighton and Sussex Medical School has documented across multiple studies that interoceptive signal strength correlates with decision quality in ambiguous situations. Anil Seth in Being You, published in 2021, frames it even more fundamentally, describing interoception as the foundation of the sense of self and suggesting that the feeling of being alive is the feeling of your body predicting its own internal state. Interoceptive accuracy is measurable. Researchers use a heartbeat detection task, asking participants to count their own heartbeats over a set interval without touching their pulse. Most untrained people perform near chance. Long-term meditators perform significantly above chance. The insular cortex itself, the primary processing hub for all of this, is one of the most evolutionarily ancient cortical regions, and its functional disruption appears consistently in the clinical literature on depression, anxiety disorders, and eating disorders across multiple research groups. The mainstream view acknowledges the body's homeostatic signals. Hunger, thirst, pain, temperature regulation, these are well-established inputs the brain uses to maintain survival. They operate largely below conscious threshold in healthy individuals, and the system is understood to function automatically without requiring deliberate attention. But Damasio's patients were not failing at survival. They could feel hunger, avoid pain, and regulate temperature perfectly. What they could not access was the subtler, continuous signal, the low-grade somatic coloring of every moment because that specific channel had been severed. And the research on interoceptive training shows the channel is not fixed. It is plastic. It improves with practice, which means it is a skill, one that is taught nowhere in the standard curriculum. What if the experience so many people describe as being disconnected from their emotions is not psychological but perceptual, the body sending a signal the mind was never trained to receive? What if the interventions most reliably shown to build emotional regulation, meditation, somatic therapy, mindful movement work precisely because they are interoceptive training in disguise? And what if that training, rather than being a therapeutic specialty reserved for people in crisis, belongs in the same curriculum as reading? From a signal you can train yourself to hear, we move to one so ancient and so powerful that when it fires, it overrides every other system in your body. And most people, in the moment it matters most, have never been told it exists. The response that freezes you and why the world got it wrong. Every story you were ever told about danger ends with action. The hero fights, the survivor runs. That is the grammar of threat, the shape of every cautionary tale ever passed between humans. But under certain conditions, sudden, overwhelming, inescapable threat, the human nervous system does something the stories forgot to include. It shuts everything down. You are conscious. You are aware of every detail of what is happening around you. You cannot move. And afterward, the world looks at you and asks why you didn't do something. Peter Levine, founder of the Somatic Experiencing Institute, spent years observing prey animals in the wild. He noticed that when caught by a predator, they regularly entered states of complete motor immobility, then shook, discharged the activation, and returned to normal functioning within minutes. Humans, he observed, enter the same state, but humans frequently do not discharge it. The nervous system stays frozen in a partial activation that can persist for years. He formalized this framework in Waking the Tiger in 1997. Stephen Porges at the University of North Carolina then gave the mechanism a precise neural address. His polyvagal theory, published in Psychophysiology in 1994, identified the dorsal vagal complex as the specific circuit responsible. A structure so ancient it predates the mammalian nervous system, shared with reptiles, capable of overriding every learned response and every trained instinct in under a second. The clinical prevalence of this response is not a theoretical curiosity. In 2017, Anna Muller and colleagues at the Karolinska Institute surveyed 298 women presenting to emergency care after sexual assault. 70% reported significant tonic immobility during the assault. 48% reported extreme tonic immobility. That data directly informed Swedish legal reform the same year. Bessel van der Kolk documented the physiological signature in The Body Keeps the Score in 2014. During tonic immobility, heart rate paradoxically drops as the dorsal vagal brake suppresses sympathetic activation. This is the opposite of the fear-based fight or flight signature. It is a different and older system overriding the newer one. Military research has found the same thing. Trained soldiers report paralysis under first contact ambush conditions even after extensive fight-or-flight conditioning because the ancient dorsal vagal system takes hierarchical priority. And the research has documented something perhaps equally damaging. The shame response that follows freeze, the years of asking, "Why didn't I do something?" is itself a secondary traumatizing agent that extends and deepens the original neurological disruption. Mainstream science would say that freeze is best understood as a delayed or inhibited activation of the sympathetic system, essentially a stunned version of the standard threat response. Dissociation during overwhelming events is a recognized psychological defense mechanism that may accompany it. These explanations are not wrong exactly. But van der Kolk's physiological data shows heart rate dropping during freeze, the dorsal vagal brake actively suppressing sympathetic activation. That is not inhibited fight or flight. That is a different system winning. The polyvagal hierarchy, social engagement first, sympathetic fight or flight second, dorsal vagal freeze as the last resort, has been confirmed in animal models and human physiological measurement. And when courts use the older, incomplete account to assess the credibility of survivors, the consequences are not theoretical. What if the single most consequential piece of missing education in human development is simply the sentence, "There is a third response. It is older than your mammalian brain. It is not a choice, and it is not a failure"? What if the shame that follows freeze is doing more long-term neurological damage than the original event? And what if discharging the activation, rather than narrating the trauma in talk therapy, is closer to the actual mechanism of recovery? Some researchers in somatic and trauma-focused therapy believe so. According to Levine's clinical framework, the body knows how to complete the cycle, it just needs permission. From the ancient brainstem circuit we share with reptiles, we move downward, into the abdomen, where a second nervous system has been running its own parallel operation since before you were born. The brain in your stomach. Somewhere in the span of your formal education, a diagram of the human nervous system was probably placed in front of you. Brain at the top, spinal cord running down, peripheral nerves fanning outward. Clean, hierarchical, the brain in command of everything below it. What that diagram almost certainly did not show was the network of 500 million neurons embedded in the walls of your gastrointestinal tract, operating independently of your skull, producing the majority of your body's serotonin and communicating with your brain primarily by talking upward rather than listening downward. Michael Gershon at Columbia University spent 30 years studying what he eventually named the second brain. His foundational discovery, formalized in the book of that name in 1998, was structural. When the vagus nerve, the primary communication cable between gut and brain, is completely severed, gut function continues entirely normally. The enteric nervous system does not need the central nervous system's permission to operate. It has its own sensory neurons, its own motor neurons, its own interneurons, its own reflexes. It is not a peripheral organ receiving instructions from headquarters. It is an autonomous system sending reports that headquarters is largely obliged to read. Emmer and Mayer at UCLA documented in the Mind-Gut Connection in 2016 that the vagus nerve carries signals approximately 80% upward from gut to brain and only 20% downward. The gut is primarily informing the brain, not the other way around. What those signals carry is substantial. The enteric nervous system produces approximately 95% of the body's serotonin and approximately 50% of its dopamine. The gut also houses approximately 70% of the immune system and a microbiome of roughly 38 trillion microbial cells that produce neurotransmitters, influence hormones, and have measurable causal effects on mood and behavior. John Cryan and Ted Dinan at University College Cork published research in PNAS in 2011 showing that germ-free mice raised without a gut microbiome display significantly elevated anxiety behaviors, and that colonizing them with microbiota from calm mouse strains measurably reduces that anxiety. Peter Holzer at the Medical University of Graz has documented across multiple studies that the gut microbiome produces GABA, serotonin, dopamine, and short-chain fatty acids that cross the blood-brain barrier. And Vallis, Colomer, and colleagues published in Nature Microbiology in 2019 that transplanting gut microbiota from depressed humans into germ-free rats induced depressive behaviors in those rats. The transplant changed the mind. Mainstream medicine acknowledges the gut-brain connection and recognizes its bidirectional nature. The brain-gut axis is understood to influence appetite, nausea, and stress responses. The gut microbiome is an active area of research. The gut-brain relationship is real and clinically relevant. But the 80-20 upward communication ratio means the gut is not primarily listening to the brain. And the Valles-Colomer microbiota transplant is not a correlation. Transplanting a depressed person's gut bacteria into a rat and producing measurable depressive behavior is a causal claim. The gut is not influencing mood around the edges. In some measurable respects, according to the evidence currently available, it appears to be generating it. What if the most effective frontier in mood disorder treatment in the next generation turns out to be gastrointestinal rather than neurological? What if the experience of a bad gut feeling is not the brain projecting anxiety downward into the digestive system, but the enteric nervous system filing an accurate upward report that the brain is misattributing to circumstance? And what if the near-total absence of gut health education in schools is producing generations of people adjusting brain chemistry while leaving the organ that makes most of it entirely unaddressed? Some researchers in neurogastroenterology are beginning to ask exactly those questions. Whether the answers reshape standard psychiatry remains, for now, an open question. The signal running continuously inside your own body is one thing, but there is a signal your body reads from other people without consulting you through a mechanism so precise it functions as an involuntary empathy test you have been taking your entire life without knowing the subject. The empathy test you've been taking your whole life. You have read the word yawning several times now. Something may have shifted in your jaw. That is not a quirk of reading, not a trick of attention. According to the research, it is a measurable index of your capacity to model another person's internal state, and the people in your life who seem completely immune to it, sitting unmoved while everyone around them catches the wave, are providing neurological information that is worth knowing. Psychologist Molly Helt at the University of Connecticut was studying children with autism spectrum disorder in 2007 when she noticed a consistent asymmetry. Typically, developing children caught yawns from adults and peers at high rates. Children on the autism spectrum showed significantly reduced contagious yawning, approximately 11% compared to approximately 43% in typically developing peers. Her paper in Child Development in 2010 was the first to formally document the link between contagious yawning and empathic processing. What followed was a convergence of findings from multiple research groups that painted an increasingly specific picture. Andrew Gallup and Brian Swartwood published in Animal Cognition in 2012 that contagious yawning rates are higher between individuals with stronger social bonds, strangers yawning contagiously at significantly lower rates than friends and family. Brian Rundle, Matthew Vaughn, and Michael Stanford published in Personality and Individual Differences in 2015 that individuals scoring higher on psychopathy measures show significantly reduced contagious yawning. Ivan Norcia and Elisabetta Palagi documented in PLOS 1 in 2011 that the frequency of contagious yawning in humans follows the gradient of emotional closeness, highest between kin, lowest between strangers. Teresa Romero and colleagues published in PLOS 1 in 2013 that dogs catch yawns from their owners at significantly higher rates than from strangers, tracking the gradient of emotional bond rather than mere species membership or co-presence. EEG studies have found that contagious yawning activates the mirror neuron network and the right hemisphere temporal parietal junction, a region specifically implicated in theory of mind, the capacity to model another person's internal state. What is perhaps most remarkable is that the physiological mechanism of contagious yawning remains genuinely unresolved. No thermoregulation hypothesis, no brain cooling theory, no oxygen regulation model accounts for the social-emotional gradient. The why is still open. Mainstream accounts suggest that contagious yawning is a social synchronization mechanism aligning a group's collective arousal level. It is understood to relate to attention and shared brain state, and the relationship to empathy is acknowledged as interesting, but not necessarily causal. But social synchronization doesn't explain why the response follows the gradient of emotional closeness rather than the gradient of simple co-presence. If it were aligning group arousal, strangers in the same room should yawn contagiously at similar rates to close family. They don't, and the gap is measurable and replicable across cultures and species. The gradient matches empathic investment, not proximity, and that match has never been satisfactorily explained by the synchronization account. What if contagious yawning is a passive, involuntary readout of the same neural system that produces compassion, social bonding, and the ability to feel what another person is feeling? What if the people who consistently fail to catch your yawns are telling you something about their empathic engagement that they would never consciously report? And what if the fact that this signal is involuntary and impossible to fake means your body has been running an empathy audit on everyone around you every day without ever informing your conscious mind of the results? According to the current evidence, that audit has been running since you were old enough to be in a room with other people. From the social signal your body broadcasts, we move to a discovery made in the laboratories of Stanford that reframes one of the most damaging public health narratives of the last 50 years: the stress that heals and the stress that kills. A study tracked 30,000 Americans for 8 years. At the start, researchers asked each participant 2 questions. How much stress have you experienced in the past year, and do you believe stress is harmful to your health? The results, published by Keller and colleagues in Health Psychology in 2012, were striking. People who reported high stress had a 43% increased risk of dying during the study period, but only if they believed stress was harmful to their health. People who reported equally high stress but did not believe it was harmful had the lowest mortality risk of anyone in the study, including people who reported very little stress. The stress was not killing them. The belief about the stress was. The mechanism behind this finding was being mapped simultaneously in sports and performance psychology. Mark Seary at the University at Buffalo had spent years measuring what happens inside the body during different kinds of stress using precise cardiovascular assessment. His research showed that the same objective stressor produces 2 physiologically distinct responses depending on how the person appraises it. Threat stress, the appraisal that you may not have the resources to meet the demand, elevates cortisol and constricts blood vessels. Challenge stress, the appraisal that this demands a lot and you can meet it, elevates cortisol and DHEA simultaneously. DHEA, dehydroepiandrosterone, is neuroprotective, and it counteracts some of cortisol's neurotoxic effects. The cortisol to DHEA ratio is used clinically as a biomarker of physiological resilience. Challenge stress also produces a cardiovascular response closer to aerobic exercise, increased cardiac output without vasoconstriction. The 2 states are measurably different at the level of blood vessel behavior. Alia Crum at Stanford brought this together with the belief component. In research published with Peter Salavi and Sean Achor in the Journal of Personality and Social Psychology in 2013, a 2-minute mindset shift video shown before a stressful task produced a measurable shift from threat to challenge physiological profile. The body followed the framing. James Gross at Stanford has documented in his emotion regulation research that cognitive reappraisal, changing the interpretation of a situation rather than suppressing the response, produces measurable changes in amygdala activation and autonomic nervous system output, and that with practice, the change becomes automatic. The long-term structural cost of getting this wrong is documented in Robert Sapolsky's work published in Why Zebras Don't Get Ulcers in 2004. Chronic threat stress is associated with hippocampal volume reduction. Challenge stress shows no equivalent structural effect in the current literature. Mainstream science would say that stress is a physiological arousal response to perceived demand, that chronic stress is harmful due to the cumulative effects of cortisol, and that managing stress means reducing exposure or developing coping strategies to limit the physiological response. This is not wrong, but the Keller data doesn't show that high-stress believers in harmlessness simply cope better. It shows their biology is measurably different. The same quantity of stress producing different hormonal ratios based on a stored belief is not attitude. It is the brain's prediction model directly regulating the endocrine system. The belief is upstream of the biology. What if the most damaging element of the message, stress kills, reduce your stress, is that it functions as a nocebo, a harmful belief that makes the stress more lethal for the people who hear it most? What if generations of people were handed a narrative that converted their normal challenge stress into threat stress and then handed medication for the downstream symptoms? And what if the most evidence-supported intervention available, teaching people to appraise demand as challenge rather than threat, is also the cheapest, the simplest, and the most consistently absent from the places where people actually learn things? Some researchers believe the answer to all three questions is yes. Whether institutions will act on that belief is, supposedly, a different question. From the signal your brain generates about your circumstances, we move to the signal your body reads from the physical world around you, a signal so precise it changes your judgment of other people, based on the temperature of an object you held for 30 seconds. The temperature of the person in the room. In 2008, researchers at Yale handed people either a warm cup of coffee or a cold drink, briefly, casually, in the hallway on the way to what participants were told was the actual experiment. Then they asked those participants to read a brief description of a stranger and rate that person's personality. The people who had held the warm cup rated the stranger as significantly warmer, more generous, and more trustworthy. The people who had held the cold drink did not. They had no idea what had happened. Their judgment of another human being's character had been calibrated by the temperature of a beverage they held for 30 seconds. Lawrence Williams and John Bargh published that study in Science in 2008, and it became the empirical anchor for a field that had been building theoretically since George Lakoff and Mark Johnson proposed in Philosophy in the Flesh in 1999 that abstract concepts, social warmth, moral purity, psychological weight, share neural substrate with the physical sensations that gave them their metaphorical names. The metaphors, they argued, were not decorative, they were structural. The brain imaging data that followed showed they were right. The temperature study was not isolated. Chen-Bo Zhang and Katie Lillienquist published in Science in 2006 what became known as the Macbeth effect. Participants who recalled a past moral transgression expressed stronger desire to physically clean themselves, and those who then washed their hands showed measurably reduced moral guilt. Bargh and colleagues published in Psychological Science in 2008 that physical warmth priming increased prosocial behavior, with warm cup participants more likely to choose a gift for a friend rather than themselves. Naomi Eisenberger, Matthew Lieberman, and Kipling Williams published in Science in 2003 that social exclusion, being excluded from a computer game, activated the dorsal anterior cingulate cortex, the same region that activates during physical pain. Joshua Ackerman, Christopher Nossera, and John Barg published in Science in 2010 that holding a heavier clipboard made participants rate job candidates as more serious and social issues as more important. Antonio Damasio's work identifies the insular cortex as the primary convergence zone for both interoceptive signals and social-emotional processing, suggesting physical and social sensation are not two systems sharing a metaphor, but one system doing two jobs. Cross-cultural replication across American, European, and Asian populations has shown a consistent directional effect, confirming this is not an artifact of English metaphor or Western association. The mainstream account describes these as priming effects, temporary context, dependent influences on judgment operating through associative memory. The warmth-warmth association was learned through repeated co-occurrence of warm bodies and warm social environments. Physical sensation activates the associated concept through spreading activation in semantic memory networks. These effects are real but modest. But spreading semantic activation would show up in language processing regions. The neuroimaging shows activity in the insular cortex itself, a region that processes physical sensation directly, not linguistically. The overlap is not conceptual, it is anatomical. And if the overlap is anatomical, then every physical environment you enter is running an unconscious calibration on your social and moral cognition before you form a single conscious judgment about the people inside it. What if the design of high-stakes physical environments, courtrooms, hospital consultation rooms, examination halls, interrogation suites is doing something to human judgment that no architect, no lawyer, and no educator has formally audited? What if the ambient temperature of the spaces where the most consequential decisions are made is part of the outcome, invisibly, consistently? And what if something as simple as the warmth of a surface is measurably reducing the probability of being trusted in a room? Some researchers in embodied cognition believe the effects are large enough to warrant exactly that audit. Whether institutions have yet commissioned it is, according to the available record, a different story. From the way the external world calibrates your social judgment without your awareness, we move inward to the state your brain enters when it appears to be doing absolutely nothing, which turns out to be the most expensive operation in your entire nervous system. The cost of doing nothing. In 1997, a neuroscientist noticed something wrong with his data. Between tasks, during the rest periods when participants were told to simply wait, a specific network of brain regions did not go quiet. It lit up, the same regions in the same pattern in every participant every time. The brain was working harder at rest than it had been during the task it was given. Nobody had predicted this. Nobody had a name for it. And when researchers finally understood what the brain was doing in those moments, the answer reframed distraction, creativity, identity, and the school system's centuries-long war on the wandering mind. Marcus Rachel and colleagues at Washington University in St. Louis documented the default mode network through PET imaging studies and published the foundational paper in PNAS in 2001. The network had been cataloged previously only as task negative, defined by what it was not doing. Rachel argued it should be understood as a system with its own distinct functions and its own metabolic signature. In his 2010 paper, in Nature Reviews Neuroscience, he documented that the resting state default mode network accounts for approximately 60 to 80% of the brain's total energy use. More energy than almost any focused task demands. Randy Buckner, Jessica Andrews Hanna, and Daniel Schachter published a comprehensive review in the Annals of the New York Academy of Sciences in 2008, linking the default mode network to autobiographical memory, future simulation, social cognition, and narrative self-construction. Jonathan Schooler at UC Santa Barbara has documented that mind-wandering episodes are disproportionately associated with subsequent creative insight, what researchers call the incubation effect, where solutions to problems emerge after, not during, periods of deliberate focused attention. Matthew Killingsworth and Daniel Gilbert published in Science in 2010 the results of an experience sampling study with 2,250 adults across 83 countries, finding that people spend approximately 47% of waking hours not thinking about what they are actually doing. Mind-wandering in this data was associated with future planning, not cognitive failure. The default mode network's significance extends into structural vulnerability as well. Buckner and colleagues documented in 2005 that amyloid plaque deposition in Alzheimer's disease targets the default mode network before any other brain region, suggesting its integrity is foundational to identity and social cognition. Research on children diagnosed with ADHD has found reduced default mode network suppression during tasks, suggesting the core difficulty may be a switching deficit rather than a deficit in the network itself. The mainstream view holds that the default mode is a resting state network handling internally directed cognition, that mind-wandering during tasks represents attentional failure and measurably reduces task performance, and that the educational goal of sustained attention to external material is neurologically grounded. These are not unreasonable positions, but a system consuming 60 to 80% of the brain's metabolic resources is not resting. It is running the most energy-intensive operation in the central nervous system. And the association between this network's activity and creativity, future simulation, and social modeling suggests that what appears as distraction from the outside is, from inside the biology, the brain constructing the architecture of personhood. What if the most valuable cognitive work a student performs during a school day happens during the moments they appear to be absent? What if unscheduled, undirected time, structured boredom that allows the default mode to run fully is not a failure of the educational environment, but a precondition for the creativity and social intelligence the educational environment claims to produce? And what if the attention economy, the notification, the feed, the unending external demand is not merely distracting people, but specifically and chronically starving the one neural system that builds the self? Some researchers in cognitive neuroscience and developmental psychology believe the implications are serious enough to constitute an educational emergency. Whether that framing has reached school design committees is reportedly still a work in progress. From the network the brain runs, when you appear to be doing nothing, we move to a system running every individual cell in your body on a genetic clock that received the Nobel Prize in 2017 and has still not changed a single school's start time. The clock in every cell. In October of 2017, the Nobel Prize in Physiology or Medicine was awarded to Jeffrey Hall at the University of Maine, Michael Rosbash at Brandeis University, and Michael Young at Rockefeller University for discovering that every cell in the human body contains a 24-hour genetic clock, not a sleep preference, not a neurological rhythm housed in the brain. A molecular feedback loop running in every cell, in every organ, in every tissue, coordinating the most fundamental biological operations according to time of day. Your liver cells know what time it is. Your immune cells know what time it is. The genes that repair your DNA run on a schedule. And every morning, hundreds of millions of students wake up at a time that is measurably incompatible with the clock their genes are running. The world gave it a Nobel Prize, then kept the school start times the same. Hall, Rosbash, and Young spent decades isolating the molecular mechanism, the period gene, the timeless gene, and the protein complexes they produce in a self-sustaining 24-hour oscillation. Their work established that circadian rhythms are not a neurological phenomenon located solely in the brain's suprachiasmatic nucleus. They are a cellular phenomenon. Every cell carries this clock. Chronobiologist Till Roenneberg at Ludwig Maximilian University of Munich translated the molecular findings into population-level data. In a chronotype study across 65,000 participants published in Current Biology in 2012, he documented that approximately 69% of the European population has a chronotype misaligned with standard social schedules. The adolescent phase delay, the biological shift that pushes natural sleep and wake times approximately 2 hours later at puberty, was documented as universal across his dataset, appearing at puberty and reversing in the mid-20s in a pattern that tracks developmental biology and nothing else. The clinical implications have been formalized at the institutional level. Judith Owens and colleagues published a policy statement in Pediatrics in 2014 on behalf of the American Academy of Pediatrics recommending that middle and high schools start no earlier than 8:30 in the morning on the basis of documented adolescent phase delay. Fewer than 20% of United States schools comply. Anna Phillips and colleagues published in Vaccine in 2016 that natural killer cell activity peaks in the morning, and vaccine immune response is measurably stronger when administered in the morning, demonstrating that the circadian clock regulates immune function with practical medical significance. DNA repair enzymes also operate on a circadian schedule, and the World Health Organization classified shift work as a probable carcinogen in Group 2A in 2007 because disrupting the clock increases cancer risk. Sutton and colleagues published in Cell Metabolism in 2018 that the same meal consumed at 8 in the morning versus 8 in the evening produces measurably different blood glucose responses. Eating against the clock is an independent metabolic risk factor. Matthew Walker documented the cognitive, immune, metabolic, and cardiovascular consequences of circadian disruption in Why We Sleep in 2017, describing chronic sleep deprivation in adolescents driven by early school start times as a public health emergency affecting hundreds of millions of young people. Mainstream explanations attribute adolescent late sleep patterns significantly to social habits, screen use, irregular schedules, and evening activity. School start times are acknowledged as shaped by logistical constraints, including bus scheduling, working parents' needs, and staff contracts. Sleep hygiene education is offered as a practical intervention within existing structures. These constraints are real, but Roenneberg's data is not self-reported preference. It is the largest chronotype dataset ever assembled, and the adolescent phase delay appears at puberty and reverses in the mid-20s in a curve that matches no social habit and every developmental biology timeline. The phase delay preceded smartphones. Screens worsen it. They did not create it. And sleep hygiene cannot override a genetic clock confirmed by a Nobel Prize. What if the global adolescent learning and mental health crisis is not primarily a curriculum problem, a motivation problem, or a technology problem, but a timing problem? What if generations of students have been assessed on their cognitive capacity during the biological equivalent of the middle of the night? And what if the most evidence-based, Nobel-supported, pediatrically endorsed intervention available has been repeatedly declined, not because the evidence is insufficient, but because the logistical inconvenience is real and the political will is not? Some researchers describe it as one of the most thoroughly documented and most thoroughly ignored findings in educational biology. We may eventually find out whether they are right. From a system governing every cell in your body on a genetic schedule, we make one final descent to the deepest and strangest signal of all. the one that reveals that pain and suffering are not the same thing, that they run on different circuits, and that one of them is more modifiable than almost anyone was ever told. The person who felt pain and didn't mind. In a neurological ward, a patient is asked to rate a painful stimulus. They answer accurately. Sharp, 7 out of 10. Left hand, localized. Their sensory system is intact. Their measurement is precise. Then the researcher asks, "Does it bother you?" The patient considers. "Not really," they say, without distress, without drama, without any of the behavioral signatures of a person in pain. They are not numb. They are not dissociated. They feel everything exactly as it is. They simply do not suffer it. And what that single clinical observation reveals about the architecture of human experience is one of the most quietly consequential things in the neuroscience literature. Physician and researcher Paul Brand spent decades documenting pain asymbolia cases and published his clinical framework in The Gift of Pain in 1997, formally articulating the distinction between nociception, the sensory measurement of damage, and suffering, the effective response to that measurement. Ulf Baumgartner and colleagues at the German Research Center of Pain published experimental documentation of asymbolia in insular lesion patients in pain in 2006, confirming intact and accurate sensory rating alongside an absent aversive component. The neural address is specific: damage to the anterior insular cortex and anterior cingulate cortex. These same structures, when damaged, also produce asymbolia for disgust, identifying this region as the convergence point for "this matters negatively to me" across both physical and social modalities. The two primary aversive interoceptive signals, pain and disgust, share a neural home. The meditative parallel was then documented through neuroimaging. Antoine Lutz, John Dunn, and Richard Davidson at the University of Wisconsin-Madison published in PNAS in 2008 that long-term meditators with more than 10,000 hours of practice show reduced coupling between anticipation of pain and pain experience, with anterior insula activation patterns measurably different from novice meditators. Joshua Grant and colleagues published in Cerebral Cortex in 2010 that Zen meditators show measurably thicker gray matter in pain-related brain regions and reduced pain unpleasantness ratings without equivalent reduction in pain intensity ratings. The sensory affective decoupling was visible not just in function, but in anatomy. Fadel and colleagues published in the Journal of Neuroscience in 2011 that 4 days of mindfulness training, 20 minutes per day, produced a 57% reduction in pain unpleasantness ratings and a 40% reduction in pain intensity ratings, with insular and anterior cingulate changes documented on neuroimaging. The Buddhist framework had named the underlying structure centuries earlier. The first arrow is the sensation. The second arrow is the suffering about the sensation. And the second arrow, unlike the first, was described as optional. Mainstream medicine holds that pain is a unified warning signal whose aversive component is inseparable from its sensory component under normal conditions because the aversion is the evolutionary purpose. Without distress, damage would not be avoided. The asymbolia condition is a neurological deficit. The meditative effects are real but modest, best understood as attention modulation rather than structural alteration of the pain system. These are reasonable positions. But Grant's meditators were not losing protective function. They withdrew from tissue-damaging stimuli appropriately and maintained normal reflexes. What changed was suffering, not detection. And if 4 days of 20-minute sessions produces a 57% reduction in pain unpleasantness through measurable changes in insular and anterior cingulate activation, the neural circuit linking sensation to suffering is not immutable. It is a learned linkage. And learned linkages, under the right conditions, can reportedly be partially unlearned. What if the most important piece of missing information in human pain education, and perhaps in all education about suffering, is the anatomical fact that sensation and suffering run on different circuits, and the second circuit is modifiable in a way the first is not? What if the question, how much does it hurt, and the question, how much does it matter that it hurts, have different answers, different neural addresses, and different degrees of trainability, and that distinguishing between them is a skill that belongs in every life rather than in specialized traditions? And what if the second arrow, the suffering about the suffering, the anxiety about the anxiety, the depression about the depression, is where the majority of chronic human pain actually lives? Some researchers in contemplative neuroscience and clinical psychology believe we are only beginning to map the practical implications of that question. 10 signals, all documented, all running right now, in this body, in this moment, without waiting for permission. The brain forecasting rather than reacting. The gut reporting upward rather than receiving instructions downward. The nervous system freezing rather than fighting or fleeing. The clock in every cell running on a schedule that morning alarms routinely violate. The jaw measuring other people's empathic engagement without announcing its findings. The stress hormone ratio being set not by circumstances but by a stored belief about those circumstances. The warmth calibration. The temperature of your last beverage quietly performed on your judgment of the next person you met. The most metabolically expensive operation in your nervous system firing precisely when you appear to be doing nothing. The sensory affective circuit separating pain from suffering and responding to 4 days of practice. These are not fringe findings. They are peer-reviewed, Nobel-acknowledged, institutionally confirmed pieces of human operating architecture, and they were left out of the manual almost everyone was handed. The mainstream view has a fair answer for why. Curricula are designed for coverage, not for cutting edges. The timeline for translating research into standard education is long, and some of these findings are still evolving at the edges. Not everything here has achieved full consensus, and the institutions responsible for education are managing hundreds of competing priorities simultaneously. That is a reasonable position, and yet some of these findings have had their Nobel Prizes for years. Some carry American Academy of Pediatrics recommendations. Some have been replicated across dozens of studies in multiple countries over multiple decades. At some point, the gap between what the science says and what schools teach is no longer a question of timing. It becomes a question of priority. The philosophical dimension is perhaps the most uncomfortable part. The body has been communicating in a complete language this entire time. It sends signals about what it needs, what it fears, what other people are feeling, what time it is at the cellular level, and whether the suffering you are experiencing is attached to a sensation or to a belief about a sensation. Every one of those signals was running before anyone named it. The naming does not create the signal. It changes what you can do with it. Most people finish a formal education and walk away with no framework for reading the organism they live inside. They navigate their emotions without knowing they are forecasts. They make decisions without knowing their gut is upstream of their reasoning. They experience freeze and call it failure. They lose sleep to a genetic clock no one mentioned. They suffer a second arrow they were never told was optional. You now have 10 words of the language. That is 10 more than most people carry out of school. Go to the comments and tell us which of these 10 signals explains something that happened to you before you had a name for it. Not an analysis, just what it was. One signal, one moment. The comments are going to be extraordinary. And if you want the next 10 words of the language, subscribe. This is where the rest of the manual lives. Your body has been sending signals your entire life. The gut filing reports, the clock in every cell keeping time, the jaw quietly taking attendance on everyone's empathy, the anterior insula deciding every moment whether a sensation becomes a wound. You did not have the vocabulary, now you have 10 words of it. Go back to where we started. The brain that was already forecasting before you thought your first thought this morning. It was running then. It is running now. It has never stopped. Your body was never the mystery. The silence was.